Okay, it looks like most people who are intending to attend have attended. So hello everyone, welcome to another Physiological Society scientific theme webinar. This one's from the Human and Environmental and Exercise Physiology Special Interest Group, titled Stimulating Skeletal Muscle Protein Synthesis and whether the quality of isolated protein is important when it's ingested with other nutrients. So just before I introduce our speakers, I have a couple of housekeeping comments to make. So firstly, our speakers are going to present. They're going to each deliver a 20-minute presentation, and then they'll present straight after one another, and then we'll have a 15-minute session for questions and answers afterwards that myself and my co-chair, Dr. Kostas Tsintzas, will chair. We have a question and answer session, a question and answer tab for people to put your questions up. So you can write those questions at any time, and they'll be directed to the speakers during the question and answer portion of the webinar. It would be helpful if you have one of the specific speakers to direct your question to, if you could actually write on your question who you would like that directed to, and you could equally say directed to both speakers as well if you would like. You have the option to submit questions anonymously if you'd like to. If not, then please include your institution at the beginning of your questions that I can announce this with your name. We might not have time for all of the questions, but we'll try and get through as many as possible, and we'll probably just run through them in order that they've been asked. Also, please note that this webinar has been recorded and will be available to rewatch. So on to our speakers. Our first speaker is Dr. Nicholas Burrs. He's an associate professor from the University of Illinois in the Nutrition and Exercise Performance Research Group. His talk is mainly going to be focused on looking at how animal protein foods affect the regulation of muscle protein turnover. And then we're going to move on to our second speaker, Dr. Benjamin Wall, who's an associate professor of nutritional physiology from the University of Exeter. And his talk is also going to focus on muscle protein turnover, but look at protein sources derived from non-animal sources. The reason for this and the reason for setting up this seminar is that we've known for a long time that there's a synergistic effect of exercise and nutrition on skeletal muscle protein synthesis. And this provides the acute physiological basis for how hypertrophy occurs in response to prolonged resistance exercise, but also how we can lose muscle mass in disease states. We've known this for some time. This is all some excellent research that's mainly come out of the Journal of Physiology and other very good physiology journals. But the majority of this research to date has tended to use, from a nutritional point of view, protein sources that are isolated or have been given, or isolated from a whole food source that have been given in isolation. So there's been some very interesting research that's come out recently to suggest that when protein is ingested as a whole food with other nutrients and potentially other macronutrients in there as well, then this can have perhaps a more prominent effect of stimulating muscle protein synthesis than when the protein is ingested in isolation. So the reason for inviting Nick and Ben is that they're probably the leaders in this field at the moment, and particularly from Nick with animal protein sources and Ben with non-animal protein sources. And there's also perhaps a contentious issue in the literature around whether animal protein sources can stimulate muscle protein synthesis to a greater degree than non-animal protein sources. So hopefully, once Nick and Ben have spoken, we may be able to have some sort of discussion around that as well. So enough from me. I'm now going to move on to and allow Nick to speak. As I've said, after Nick has spoken, Ben will then speak, and after that we'll have a Q&A session. The only thing from me to say is thank you ever so much to everyone who is attending today. I think we've had a very good turnout. I also think I forgot to introduce myself. I'm Professor Francis Stevens from the University of Exeter, and I will see you all in about 40 minutes when we chair the Q&A session. Over to you, Nick.
just had to find the unmute button. Deal with me. Thanks for that introduction, Francis. I appreciate that. I can see you use the word leader pretty loosely over there, but I'll give it my best shot here. Just a brief outline that I want to cover today. Just as a backdrop, first provide a basic overview of what we know about with regards to just eating amino acids. Certainly they stimulate muscle protein synthesis, and then as alluded to, transition into more recent developments related to whole animal-based protein foods. So just a brief note here that in our laboratory and many others, of course, we generally try to understand protein quality by determining how well the food we eat stimulates muscle protein turnover, but keep in mind DIAS is another protein quality score that is often used. The DIAS is based on true ileal digestibility, determined at the end of the small intestine, where amino acids are absorbed, and generally the growing pig model is going to be used. So, you know, the question is, why do we use a human muscle? Well, it's highly responsive to both nutrition and exercise. Metabolically, obviously has a number of important roles. So it is an important regulator of postprandial glucose disposal by virtue of its mass, a large site of lipid oxidation, in addition to its role for locomotion, strength, power, also a large contributor to basal metabolic rate, which for most of us is going to be the biggest contributor to total daily energy expenditure, and thus obviously has an important role in weight maintenance. And then, you know, during times of catabolic stress, it is a large reservoir of bodily amino acids. And now another important notion here is that we can provide recommendations in terms of protein requirements on a meal by meal basis, which we think is useful given the fact that perhaps meal frequency is relatively important in terms of maximizing dietary protein use throughout the day. So this is just again, just a basic overview of muscle protein turnover in response to nutrition and exercise. So what we see here in the red is going to be protein breakdown, and the other variable, of course, is synthesis. So in the resting state, we can see that protein breakdown is going to exceed synthesis. So if we do the math, we're going to be in a negative protein balance. When we eat breakfast, in particular protein-containing meal, we're going to see protein synthesis is the primary variable being regulated. So we're going to swing into a positive net protein balance. And essentially what this is going to do is to counterbalance any fasting state losses. So individuals eating adequate amounts of protein, adequate amounts of energy, skeletal muscle mass is going to remain relatively unchanged. Now, exercise is going to create a situation in the fasting state, rather. Once again, we see a strong stimulation of protein synthesis. Of course, exercise of any nature, you're going to see a stimulation of protein breakdown as well, but we are shifting more to the positive in terms of a positive net protein balance. So exercise is a fundamentally anabolic stimulus, even in the fasting state. But of course, we need to eat food to create a situation where we're in a positive protein balance. This is a situation where we're going to obviously, with repeated use of a particular exercise and feeding paradigm, that we can elicit a positive adaptation. And here we can see as well as that the exercise amino acid interaction essentially creates a synergy on muscle protein synthesis. So, you know, the major take-home point with this slide is that eating amino acids strongly stimulates muscle protein synthesis, and exercise is interactive on this response. So that largely was based on free amino acids, but we can see we can elevate amino acids via different avenues, different ways, whether it's liquid food, intravenous infusions of mixed amino acids. We know that the essential amino acids are particularly anabolic. We can give a single amino acid, eat isolated protein powders, or of course food as well. We see that they all can induce a strong stimulation of muscle protein synthesis. Now, piggybacking off of that notion is that researchers have been really curious or putting forward the notion that the amino acid content of protein-containing food is really important, in particular the leucine content. And the reason that could be true is because obviously if we're eating a high leucine containing food source. This is subsequently going to directly impact the subsequent leucinemia. And it's believed that the postprandial leucine profile is important for regulating the muscle protein synthetic response or the anabolic response, with whey being especially anabolic because, again, when we look at the amino acid composition of this food source, it's going to have a high leucine by total amino acid mass relative to soy or casein, as noted here in this figure. So when we eat soy, it's going to be rapidly digested. It's going to elicit the highest blood leucine concentration and generally a larger muscle protein synthetic response. Right. And now another important notion here with this leucine trigger or leucine threshold hypothesis is that there's not a maximal switch on, but it's probably a dose increase. So of course you can elicit a rise in muscle protein synthesis with lower leucine concentrations. So if we put our current body of evidence or knowledge together, this is sort of what we can come up with here. So what is an ideal protein containing food? What would it look like? So of course we want the amino acid composition that mirrors that of human body protein to prevent any amino acid antagonism. And basically amino acid antagonism is describing a situation where you might induce mutually adverse or opposing actions of like amino acids. So a good example of that would be the branch chain amino acids. If you fortify a food source too high with leucine, you could interfere with isoleucine or valine absorption and subsequently impact growth. So we can also leverage a food source too high in essential amino acids. This has largely been shown in agriculture, so growing chickens, these kind of things. But certainly a high leucine by total amino acid content is important. When you're eating isolated food proteins, a fast speed of digestion and absorption is relevant. This ultimately is going to help elevate the net exposure of amino acids. A lot of work has been done where we don't think that perhaps the absolute energy or in subsequent insulinemia that's induced is highly anabolic. It's more the amino acids that are driving the response. And then if we have a lot of amino acids available to tissue, muscle tissue, they can take them up in the muscle, of course, turn on some anabolic signaling pathways, and then start the process of protein synthesis, which we just learned is the primary variable dictating net protein balance throughout the day. Now, you know, it's always interesting to me when we look at the dose response curves after isolated animal proteins is that we see the stepwise increase in muscle protein synthesis until we hit an eventual plateau. And that plateau has been shown to occur roughly, just for simplicity's sake, about 20 to 25 grams of protein. But when we look at the other end of the spectrum here on oxidation, at a presumably optimal dose, we also see large rises in leucine oxidation in particular. Some time ago, I used to think maybe this was also a signal for the muscle to tell it that there's plenty of substrate available, but I think this might just simply be a product of eating isolated proteins. We can certainly elicit a large response, but at the same time, we're also elevating leucine oxidation. So I always view these dose response curves more as maximal dose response curves as opposed to optimal, which in my mind, an optimal definition would be, of course, eliciting a large rise in protein synthesis, but with minimal oxidation rates. Now, one thing I guess we need to be thoughtful about is that as we start to really show that these dose response curves based on these isolated protein sources are quite high, I think we need to also recognize that if, you know, all I'm showing here is a performance plate, but if we start to overtake a performance plate with protein, there's a chance that we're going to also displace other vital nutrients. And I think more of us are recognizing that as well. So we sort of been flipping the switch, if you will, or the paradigm a little bit. So I obviously recognizing that focusing on parts of food, amino acids or isolated protein sources was, of course, a good starting point, and it had to start there with regards to understanding how the food we eat regulates muscle protein turnover. However, you know, us and many others are attempting to understand how perhaps whole food proteins, potential food matrix interactions, and or meal combinations, which ultimately are what really make up a dietary pattern, And when we're thinking about dietary patterns, it's important to remember that they are adaptable. Here in the US, we have a US style, which a lot of us adhere to. It's going to contain a lot of animal-based food proteins. And then an adaptation of the US style is going to be a Mediterranean style. And of course, another adaptation is a vegetarian style, which I won't spend a lot of time on. I'm going to leave that for Ben to discuss. But, you know, so it's always important to keep in mind that these are adaptable. You know, and essentially what I'm trying to say is, you know, what is the best type of protein? Well, it's the kind you eat. It's going to really depend on what particular eating pattern you're adhering to. So with that said, you know, one of our first attempts was during my postdoc with Luc van Loon. We essentially started to explore this notion of whole protein foods by comparing beef and milk. And what we're looking at here is, of course, they have a fancy tool, intrinsically labeled food protein, to provide estimates of digestion and absorption rates after you ingest these food sources. And we can see that minced beef is more rapidly digested and absorbed, or results in a greater postprandial release of dietary phenylalanine when compared to milk. So a blend of whey and casein. Now, when we looked at the FSR, and this is the temporal response, so FSR is simply a direct measurement of muscle protein synthesis, we did find something quite interesting here. Despite this more rapidly digested profile of beef, which perhaps if we just looked at that alone, it would indicate that it's maybe has a little more anabolic potential. But what we found here is that milk is more anabolic than beef, at least during the short term here, during this immediate postprandial period. But then when we collapse the data and look more at the long-term response, we really don't see many differences. And then the question is, which is more relevant? Well, you know, provided the notion that most of us are going to be eating every 4 to 5 hours, I probably wouldn't expect, if we were just eating beef or milk, to see a huge impact on the adaptive response based on the 5-hour response. But of course, mechanistically, this is quite interesting that milk was greater than beef in the short term. Now, you know, we didn't really have a good idea why. There was thought that perhaps it has to relate to bioactive peptides, perhaps even the endogenous release of microRNAs. So, you know, something unique about milk and some other foods, of course, is that it's believed that they can transfer some of these bioactive compounds directly into circulation, which might have some roles on regulating muscle protein turnover. And, you know, based on that notion, through some collaborators, we have been getting interested in perhaps the role of di and tripeptide. You know, are these di or tripeptides contained in food actually making it into circulation, and then ultimately can they have a regulatory ability? Now we can see here, based on this table, where we simply analyze various protein-containing foods, isolated protein sources, and then looked at the di and peptide content, in particular dileucine or trileucine. And we can see that milk protein does have a little higher dileucine content relative to trileucine, with trileucine being relatively unabundant. So we do know that these, at least from a dipeptide standpoint, they are getting into the circulation. Now ultimately the next step would be to assess whether or not there's any regulatory role in human muscle protein turnover. So maybe a potential mechanism underpinning that greater milk response. Now, this is really cool data from Kevin Tipton, sort of a really good indication here that, hey, you know what, maybe a whole food matrix might be doing something. So what he did was a comparison of whole milk versus fat-free milk versus a isonitrogenous control, used an indirect marker here of amino acid uptake, AV balance method. It showed that whole milk resulted in a greater uptake of one particular amino acid. Now that's really interesting. So what about the whole milk? So we sort of, nobody spent a lot of work on this notion for a long time until we recently explored something similar with whole eggs, which we'll talk about in a minute. Now, I wanted to highlight these data here. So when I was doing my postdoc with Luc van Loon, he did a lot of really, really great work trying to make, in this case, casein more anabolic. And he did that by adding different nutrients to isolated casein. We added carbohydrates, we added fat, and then sometime after I left, he also looked at an experiment where he added milk serum. So trying to understand, does the some of the bioactive components that could be contained in the serum, does it have any anabolic potential on isolated protein sources? So interestingly enough, if we look at all these studies, adding single nutrients to an isolated protein source didn't augment the postprandial muscle protein synthetic response. So at that time it was like, well, maybe single nutrients are fine. Maybe that's it. Maybe it's just a matter of amino acids are the main driver. So again, going off of Kevin Tipton's earlier work, Stefan Van Fleet and myself, we had a lot of good conversations, really wanted to explore this. So we approached this food matrix interaction based on comparing whole eggs or isonitrogenous amounts of egg whites. And what we found here was that the whole egg ingestion augmented the postprandial anabolic response greater than the egg white condition. And this was after exercise in healthy young individuals. What else maybe a lot of people didn't pull away from this study is when we look at the leucine oxidation rates, we see a large rise in, I'm sorry, just the exact opposite of that. We don't see a large rise in leucine oxidation despite a large rise in the postprandial muscle protein synthetic response. And again, I think this has to do with the fact maybe we're getting some food matrix interactions, more of an optimal response. This was about 19 grams of protein. And so I really like this part of the study. Again, I think we're starting to transition to optimal. But without a mechanism, we still were scratching our heads. We looked at some of just some static traditional Western blot readouts of mTOR, P70 for BP1. We didn't see any differences on that side of things. A good colleague of mine, Dan Moore, he wanted to look under the hood a little bit more, and he actually looked at some of the mTOR co-localization results. So we know that mTOR likes to co-localize to the lysosome, and subsequently that whole complex likes to move to the sarcolemma, and this is going to be, makes a lot of sense because this is the site where amino acids are going to be coming in to the skeletal muscle. And we see that in this case, whole eggs induced a greater co-localization to the lysosome when compared to egg whites. So we do see mechanistically that perhaps there is something underpinning that greater anabolic response with whole eggs. So the logical question a lot of people would ask is, what's in the egg? I wish I could answer that because then it's easy, right? We could simply fortify an egg with one particular component and make a more anabolic egg, right? And it's easy. You just grab some chickens, throw whatever anabolic component you want to in the chicken food, the chickens are going to eat it, and now we have an anabolic egg. But I don't think it's that easy. I think it's more of an interaction. These food matrix interactions, I think there's something there. And I don't think it's just one single thing that's inducing this greater anabolic response. And that's the interesting part of a food matrix, of course. This is just pivoting back to that point, when we're comparing maximal versus optimal dosing. I think with whole foods, we can leverage these food matrix interactions to get more of an optimal response. So a large rise in postprandial muscle protein synthesis without large rises in leucine oxidation. And again, I think this is more optimal versus using more of a maximal dosing approach. So that was great. That whole egg study was pretty cool, in my opinion, and probably some other people. But nobody likes to be nice, of course, as scientists. So Dan Moore and I were talking with regards to how can we really address this food matrix question. And so, the basic question is, do food components, amino acids, behave differently in isolated form than when forming part of food structures? So we just put our heads together and let's design a study to get after this a little bit. The lead on this was Kevin Paulison. This was presented at Nutrition 2020, obviously in the virtual format. But anyway, we wanted to compare a food source that is part of a Mediterranean eating pattern, in which of course is fish or salmon in this case. So what we did from a condition standpoint is we can compare 3.5 ounces of salmon, which is approximately 150 kcal, or 20.5 grams of protein, and then the fat ratios are there as well. So we sent these the piece of salmon off to get analyzed, and then we simply remanufactured the salmon with isolated nutrients. So taking free amino acid blend along with the fat ratios there shown on your slide. We tested the hypothesis that, you know, eating salmon is going to be greater than the sum of its parts, essentially. So to do this, we recruited healthy men and women. They underwent a prime constant infusion. Once again, we looked at it after a acute bout of resistance exercise, coupled with repeated biopsies and blood draws. So here's the leucine profile. So it shouldn't be that surprising that when these free amino acids are not embedded in a food matrix, they're going to behave a little differently than an intact food matrix, as noted here. So obviously the salmon, we're not going to see quite a large rise in the blood amino acid concentrations relative to the free amino acid or the free nutrient condition. But when we look at the net exposure there, which is shown here, the net exposure of leucine over the course of the whole experiment, we can see they were quite similar. Now, perhaps one of the most interesting parts of this study was, in fact, that I had no idea, or I didn't know that salmons were born with tags. No. So this obviously is farm fresh salmon. We did buy one salmon, quite expensive, and then we cut it up to use it for the whole experiment. We had some leftovers so we could have a proper salmon barbecue after the fact. But nonetheless, when we look at the FSR data, we didn't actually induce a significantly greater rise relative to the isolated nutrient condition. But there's about a 28% difference. Some might say that when you see about a 20% difference between postprandial FSRs, perhaps that's physiologically relevant. But in this case, you know, maybe it's relevant, maybe it's not, but we certainly didn't see a difference from a statistical perspective. When we look at the temporal responses, we don't see any differences as well. So, you know, one thing we can take away from these data is the notion or the fact that, you know, the whole food's is working just as well as this free amino acid blend, despite the different leucinemia profiles as well. So just a couple closing slides here. So, you know, that was one of our first attempts to look at the salmon matrix more directly, but there's certainly a lot more food sources out there. You know, as noted, that salmon wasn't that lipid rich. I think there's something with a lipid rich food matrix. So we're starting to explore food sources with a little more lipids in their food matrices, sort of like the egg, but there's certainly others. But all this slide is noting here is that certainly the type of protein that you eat matters. But I think we got to be a little more thoughtful in terms of these food matrix interactions. Different foods, of course, could perhaps have more or less greater response on muscle protein synthesis. And you know, finally, this is also to keep in mind that when we're thinking about eating protein foods, we essentially should be eating on a continuum, eating a diverse diet consisting of both animal-based and plant-based proteins. I think this ultimately is going to make up an optimal eating approach. Like I said, I'm going to leave Ben talk more about the plant-based protein sources. But that's all I have. I certainly want to thank my research team. They do all the work here. I just get the opportunity to talk about it. I also wanted to note some of my funded sources here. I have been funded by various animal-based food producers.
Nick, that was great. Thank you ever so much. Some fascinating data in there. So we're going to move swiftly on. And Benjamin Ward from Exeter is now going to talk on animal-based protein sources before we have our Q&A session. So Ben, over to you.
Thanks very much, Francis. Do you see my screen okay? You can see it?
Yes.
Okay. Well, thank you very much. And thank you to the Physiological Society as well for putting on this webinar. I think Nick has made my job a little bit easier for this presentation, because he's essentially given the overview of the basic metabolism that we're covering today. So I've really put an overview here, which is quite straightforward. In terms of context, I've been asked to place this in the context of non-animal derived protein sources. And of course, we're increasingly interested in this from a societal point of view, based on the concerns or the increasing interest in global sustainability of food production, increased ethical concerns of how and where we get our food from. But I think perhaps more relevant to today's seminar is the fact that it offers an additional look at some of the mechanisms that Nick has talked about. So my goal today is to really do a whistle-stop tour of what we know about how plant-based proteins may or may not stimulate protein synthesis rates, and really view it through the lens of two overlapping paradigms that Nick has nicely laid out already for you. So that is essentially that the protein is the fundamental macronutrient that's stimulating protein synthesis rates, and more specifically that this is occurring based on the leucine trigger hypothesis. But I'll also move on in the same way that Nick did to consider whether or not we have any data at present that will consider whether or not any non-protein factors actually support the protein synthetic response. And with this, I mean these interactions with whole food and a whole food matrix, for instance. So as has been stated a few times today, we ride on the back of a long literature base in this field for several decades of excellent work that's established an awful lot of the mechanisms underpinning how amino acids stimulate protein synthesis rates. Much of the early work relied on intravenous infusions or the ingestion of isolated amino acid sources. And then work moved on to look at intact protein sources, such as some of the data that Nick presented. And actually the data concerning whole food or mixed food ingestion is quite minimal. When we actually then say we don't want to rely on the animal-derived protein literature, we have even less, because most of our literature comes from the protein sources derived from milk or meat or eggs, for instance. So in this space, we do have some data, and we also have a paradigm on which we can build up from. And to that effect, Stefan Corison out of Luc van Loon's lab produced a really useful paper a couple of years ago, where he had a look at what proteins were available, really. And I'll just earmark that I am talking about protein isolates at this stage. And often people view a protein isolate as something that would be 100% protein, but of course it's not commercially. And this is a basic analysis, a food analysis of how much protein is present in isolated protein sources available commercially. But it's a wide array of plant-based sources compared to a good array of animal sources, and of course human muscle, as Nick alluded to, we often use as a kind of a proxy for what we think may be useful. So there's not too much difference there. You can get the food sciences to the stage where you can actually isolate proteins from various different non-animal sources without too much issue. But it's when we start looking at what's in that protein, which amino acids constitute that protein, that the typical narrative surrounding animal proteins being more anabolic than plant-based proteins is really rooted. And you can see here that this is the proportion of amino acids within the total protein of all these isolates that exist as essential amino acids. And of course, we know that essential amino acids are doing the vast majority of the heavy lifting in the postprandial stimulation of muscle protein synthesis. And in fact, when we look at thresholds that have been suggested by WHO, FAO, expert panel on protein, we see that animal proteins always exceed that threshold of what's required in terms of the essential amino acids. And plant-based proteins can either fall above or below, with quite a lot of variability across sources. And the same is true when we hone in on this particular amino acid, leucine. And of course, the fundamental aspect of the leucine trigger hypothesis is that we know leucine occupies a fairly unique role at the molecular signaling level. It appears uniquely capable of stimulating the mTORC1 complex within the muscle tissue itself. And again, the preponderance of leucine in animal proteins here is much higher than when we compare it to plant-based proteins. But, and as I'll keep returning to, it's quite variable across different plant-based sources. So while this gives us a nice sort of framework in which to operate and view whether or not something may or may not have anabolic potential, as has been suggested in Nick's work, really, particularly for muscle physiologists or those interested in the context of healthy aging or sports nutrition, where the maintenance or reconditioning of muscle tissue is key, we do want to know what the anabolic potential actually translates to in terms of real endpoints in human physiology. And when we look at muscle protein synthesis, we don't have too much data in this space. I will do a whistle-stop tour of this, but basically what I would suggest is that we have 3 protein sources that we have good data for, and that is soy, wheat, and potato protein. But of course, this is a very small proportion of those proteins that are actually available out there. So, from a traditional standpoint, this was really the first and classical paper comparing plant and animal-based proteins, like by Jason Tang and Stu Phillips' lab. And here he's used a nice approach where you've got a whey protein as a gold standard, if you like. We know whey protein is very rich in leucine, and we know it's digested very quickly, and therefore provides that anabolic trigger, the leucine trigger. And you can see this reflected on this figure in terms of that fast and big rise in postprandial plasma leucine concentration. We also know that casein, the other protein portion involved in milk, is much more slowly digested. So of course, that's going to give you a slower profile, but it is also still quite rich in leucine. And soy, as you can see here, is lower in leucine and sort of mid-range in terms of its digestion absorption kinetics. So in terms of testing the leucine trigger hypothesis, this is perfect because you can then see how this relates to postprandial anabolism. And whey protein gave that traditional potent stimulation of protein synthesis rates, with soy sitting somewhere in between that and casein. And really this sits well with an awful lot of the literature, which when looking at isolated sources, or in fact the same sources fortified with and without leucine or the other branched chain amino acids, really does support the idea that the leucine threshold or the leucine trigger is quite predictive of postprandial muscle protein synthesis rates. So it allows us to perhaps increase our interpretation of some of those biochemical breakdowns of the protein, the basic protein structures. But some more data from Stefan Orison from Luc van Loon's lab a few years ago, firstly extended that we now had data on another extremely plentiful in the diet protein source, and that's that wheat protein, but also started to suggest that perhaps the presence of leucine solely as the mechanism regulating postprandial protein synthesis rates, that perhaps it wasn't quite as simple as that. On the left of the screen, you can see the plasma leucine kinetics again. This is a very similar approach to the previous paper that I showed, where you can actually see you have whey protein fed as a gold standard in a sufficient amount to give plenty of leucine. You see the characteristic rise, very quick, very high, more so than that of casein, which was isonitrogenous. And then also in this one, WPH stands for wheat protein hydrolysate. This is isonitrogenous, so protein matched across conditions, so therefore the wheat protein had significantly less leucine content within it. When you translate this, to some extent this does follow the leucine trigger hypothesis, and that is that those proteins with a greater leucine content, so the whey protein and the casein, stimulated protein synthesis rates to a greater extent. In fact, the wheat protein didn't result in, at 35 grams, so lower leucine content, didn't contain sufficient leucine to actually produce a measurable response. But you can already see that despite the greater leucine kinetics in this study exhibited by the whey condition, that was not more anabolic than the casein condition. And the story gets more in line with perhaps thinking beyond leucine per se, when they actually, within the same paper, they made a leucine matched comparison. So what I might mean here is they adjusted for the fact that wheat protein is lower in leucine and reasoned that if it is just to do with how much leucine you actually consume, then you can just consume more of a plant-based protein and perhaps achieve the same effect. So what you see here on the left now is a plasma leucine kinetics in response to the ingestion of 35 grams of whey protein, or a leucine matched bolus of wheat protein hydrolysate, which required, because it's lower in leucine, a 60-gram bolus. And contrary to the leucine kinetics here, what you actually see is that large 60-gram bolus of wheat protein is actually capable of stimulating a greater postprandial muscle protein synthetic response compared with the whey protein. So they speculated various mechanisms that may extend beyond the leucine trigger in this paper. But it does start to point that when we compare across sources, perhaps it doesn't become as clean as just looking at postprandial leucine kinetics. So this is where we assessed some of the data a few years ago, and this is another review from Luc van Loon's lab, this time led by Stefan van Vliet, who, as chance would have it, ended up producing and I'll compliment Nick's egg paper, seeing as he was upset that nobody did, the excellent paper from Nick Bird on egg protein that was led by Stefan van Vliet several years later. But this was actually a collection from the literature on the essential amino acid content of several plant-based sources compared to animal-based sources. And as you can see, those that we've looked at, and those that we have muscle protein synthesis data for in the literature, are towards the lower end in terms of how they're represented in essential amino acids. And there are various other sources that have not been looked at. And the one that we got particularly interested in was mycoprotein. So still lower in essential amino acids compared to animal comparators, but quite high relative to a lot of other non-animal based sources. So most people are not too familiar with what mycoprotein is. So mycoprotein is technically not a plant-based source because it's actually a fungal derived protein. And it's produced in a kind of controlled agricultural way by continuous fermentation of a fungal species known as Fusarium venenatum in large vats in North Yorkshire in England. The initial species are actually placed in the vat and fed on a continuous substrate of glucose, vitamins and minerals, and ammonia to provide the nitrogen backbone to actually produce protein. And by harvesting the product, you end up with a relatively protein-rich, and it's a nutrient-dense and something that I want to come on to, whole food, in the same way that Nick's been describing that we're talking about a whole food. And it looks a bit like this, the product. It's basically a paste, which when frozen will produce a fibrous texture. And this is a picture of it under a microscope. More similar to meat, and that's why it's been commercially exploited to be the foundation of the meat alternative products that are available under the Corn banner in the supermarket. So the important thing to point out here is this is not a protein isolate. So it's quite interesting from this topic to start considering how we might investigate this. It's a protein-rich food, but it's not a protein isolate. And what I mean by a protein-rich food is it exists at about 45% protein. There's some fat and carbohydrates in there. It's quite low. It's extremely high in fiber, another point that I want to come back to. And then if we take the same approach and get a little bit more specific about the amino acid content of mycoprotein, as I pointed out, it's quite high in essential amino acids. And this is some data we put together a few years ago in a review article. But essentially this just as a comparison of the essential amino acid content of mycoprotein versus some of the traditional animal proteins that we know are so effective in stimulating muscle protein synthesis rates. And whether we look at the essential amino acids or the total of the branched chain amino acids or in fact just honing on the leucine, it's a bit lower than when we look at an isolate like whey, which is understandable. As I said, it's not an isolate. And it compares quite well. It sits somewhere in between whey, milk and egg protein. So we reasoned that assuming that you're consuming mycoprotein in sufficient quantity to provide enough leucine, which was obviously what was the wheat paper led us to realize how clearly important this was, then we should see a robust stimulation of muscle protein synthesis rates. And we ran a couple of studies where we looked at things like the digestibility and the bioavailability to ensure that actually this was a food source that was going to be available in the circulation. And then work, it was published earlier this year, led by Alistair Montaigne, led to demonstrating that proof of concept that if we actually take a sufficient bolus of mycoprotein, can we stimulate robustly muscle protein synthesis rates? So Alistair decided to take an approach here where we said, well, how much leucine do we believe we need to stimulate protein synthesis rates? And you get a few different figures from the literature, but 2.5 grams seems to be at or close to optimal based on the dose response curves. And really the dose response of protein seems to be somewhat dictated by the leucine content of the ingested protein. So coming back to the fact this is not a protein isolate, but it's a whole food source. This requires a large bolus to be consumed. So this represents 70 grams of the mycoprotein product itself. This contains about 31.5 grams of protein. So what you would expect with a good quality protein source to be sufficient to stimulate maximally protein synthesis rates or close to maximally. And that was the primary hypothesis, was to demonstrate whether or not this would in fact confer an anabolic response. But of course, we also wanted to put this in context of the literature and make a comparison with an animal comparator. So we didn't choose to do this against the gold standard whey protein. We went more towards something that's approaching a bit more of a real life food source like milk. This is a protein isolate, of course. So to get 2.5 grams of leucine, with the isolate that we actually used, you needed about 31 grams of product. And that results in about 26.2 grams of protein. So a little bit less than the mycoprotein condition, but still what we would expect to be sufficient protein to allow a very optimal anabolic response. But this is also what I want to just draw attention to is, of course, feeding a whole food results in the presence of all these other macronutrients and what I'll come on to is the micronutrients. So the overall caloric intake within the mycoprotein condition was approximately double that of milk. And this resulted in a fairly viscous solution because you've got the high fiber, which is going to actually reduce the digestion and absorption speed. And you've got a lot of product, you've got 70 grams within that. So this is a large bolus for volunteers to consume. But as most people involved in these studies know, that consuming a protein shake during an intense stable isotope day is the least of a participant's concerns. And Alistair also took the unilateral resistance training model, where we can actually look at whether or not there's a differential response in rested and exercised muscle of the same individuals. Because obviously, as Nick said, there's quite reason to believe that regulation may differ depending on the contractile status of the muscle tissue. So the first thing to show again is viewing this through the lens of the leucine trigger hypothesis is somewhat predictably, we saw that milk and mycoprotein being leucine matched, that milk led to a quicker and greater overall rise in leucine because it's so much more quickly digested. We knew this would happen. We've seen this before. This is in line with the literature. And this led us to actually hypothesize that when leucine matched, if leucine was the primary determinant, we should see an equivalent anabolic response. If digestion absorption kinetics, and specifically the speed, was the determining factor, then we should see a greater muscle protein synthetic response from milk. But when we looked at muscle protein synthesis data, so when we look at the rise from fasting in the resting or exercise muscle that you see here to the fed condition, so this is the response to protein, whether it was not in fasted or fed, we did see the robust response in response to mycoprotein that we hypothesized. But actually this was marginally greater than that we saw in the milk protein. Again, this is contrary to how we hypothesized. So similar to Nick, we wanted to have a look. This was very sort of, it wasn't approaching the whole food question at the time. It was approaching, we approached it from the leucine threshold perspective. But we wanted to get an idea of what mechanisms might be regulating this. So as muscle physiologists, we tend to look within the muscle. We had a look at the usual suspects in terms of the phosphorylation of the mTORC1 signaling complex, and we didn't see any differential activation of this pathway across conditions. We also took a targeted gene array transcriptional approach to look at the gene expression of targeted genes within various pathways. And despite seeing interesting pathway-related changes associated with exercise and protein ingestion, of the 48 genes we looked at, only a single gene demonstrated any difference across our two protein conditions. And in fact, this gene was more concerned with the regulation of protein breakdown. So again, we didn't see any evidence here that we picked up that it was transcriptionally regulated at least. So I'm just going to spend the remainder of my time looking at how we've interpreted that and then how we've developed future studies to try to get or join the party really in terms of Nick's views on the whole food, because that's not really how we set out to look at it, but we think it may be relevant. So we could first of all view our data through the more protein related idea of regulating postprandial muscle protein synthesis rates. So we fed more protein, for instance, in the mycoprotein condition. But similar to what Nick explained, we saw an acceleration of amino acid oxidation in both of those conditions, suggesting that we had plenty of substrate, plenty of protein fed. We can't, of course, rule out other signaling roles for non-essential, for essential amino acids. We've got a lot of detail in the human literature on the role of leucine, but far less for some of the other amino acids. And we also can't entirely rule out substrate limiting amino acids. We don't quite know, for instance, which are the other essential amino acids that may be lower in certain protein sources. But this seems a less likely explanation for our initial data, at least. So what about if we try to look at it from this food first approach, where we look at this as a whole food that is what we've actually fed? And have we in fact just altered the leucine threshold such that Nick suggested that you might, by feeding in a food matrix, potentiate this response, facilitate the leucine stimulation of postprandial anabolism? Well, again, if I look at Nick's figure now on the egg protein, we've, of course, analogous to that study, we've got plenty of protein. So we've directly stimulated muscle protein synthesis. That's fine. But if I look at, for instance, the idea that cholesterol might be involved, which seems like a very good hypothesis, there's good acute and long-term data to suggest there might be a relation here between cholesterol and muscle protein metabolism. The mycoprotein is actually devoid in cholesterol virtually. And in fact, if you feed it for a number of days, you actually reduce circulating cholesterol. So this seems unlikely. It's also very low in lipids, particularly in some of the omega-3 fatty acids that have been suggested to be relevant as well. So this doesn't seem like a reasonable or satisfactory explanation for our data. The bioactive peptides, we don't know what's in it, so there's various different ideas that could be involved in there. And of course, there's the vitamins and the minerals that are present in whole foods that we know interact with various metabolic pathways. And this is why I just put together this table for today, really. What you see here is the vast array of micronutrients that are always present in most of the whole protein-rich foods that we eat. And in mycoprotein here on the left, we've got an analysis of the raw product here, and I've actually made a rudimentary analysis here from data available online with milk and beef and eggs. And you can see that we don't really know a lot about whether or not various different things that we know are metabolic cofactors in important reactions throughout metabolism may or may not be interacting with protein metabolism and may or may not explain some of the effects that we've actually discussed today. And the final thing that I just draw your attention to here is I've suggested, or I've mentioned that mycoprotein is rich in fiber, but it's rich in a particular type of fiber, which is fairly unusual within the human, or at least within the Western diet, and that's chitin and beta-glucan. And emerging data do suggest that the important signaling roles of short-chain fatty acids, of course fermentable from fiber in the gut, interact with various metabolic processes. So some of the in vitro data suggest that they are capable of sensitizing the mTOR complex to leucine. But again, this remains speculative without any real in vivo human skeletal muscle protein turnover data. So I would have liked to have been able to present some of our follow-up work on this, but COVID-19 did a good job of getting in the way of Sam West, who's leading on this work, about trying to get a bit further into the mechanism here. So he's trying to take the protein out of the mycoprotein and have a proper comparison of protein within and without its matrix, very similar to what Nick had presented with the salmon data. So to working together with Quorn Foods, he's been able to actually develop the isolate, which is quite pure. It's taken out all the carbohydrate, all the fat. There's still a bit of fiber in there, but it's predominantly now looking very much in terms of a macronutrient composition like a protein isolate. And his pilot data indicate that when you feed that to humans, it also behaves like a protein isolate with respect to the postprandial amino acid concentrations, that being a much quicker, more rapid rise in the branched chain amino acids and coming down more quicker compared to the same protein fed within its initial food matrix, which is much, much slower, of course. So that's where I'm going to leave it today. I just want to make a few thank yous. Thank you very much to all our collaborators and sponsors that makes it possible to do the work, particularly Quorn, who sponsored most of the mycoprotein work presented today. Thank you to our research group, particularly Sam and Alistair, who did the work that I've described today, and then for the Physiological Society for putting on this webinar. And thank you very much for everybody's attention.
Thank you for that, Ben. Another great talk. It's a bit weird doing these seminars without having an audience to sort of clap after each presentation. It's all a bit awkward and quiet. So in the interest of time, I think we'll just dive straight onto some questions from the audience. So this is the first one for Nick. This is from Carl Friedel. Did you evaluate a possible effect of circulating humoral substances, growth hormone, IGF-1, anabolic steroids in the milk experiments. And I guess I would probably just like to add to that as well, in terms of humoral substances, any sort of incretin effects perhaps, or some other gut-derived peptides.
In the milk experiments with Luc Van Loon, no, we didn't evaluate any of those other aspects. Generally, from all the great work Stu Phillips has done, at least in terms of circulating IGF-1 and testosterone, we don't see a big impact directly on muscles. More of an intrinsically regulated process, but certainly there's a host of other things you could evaluate. I agree, and we have some other experiments going on where we are probing in a little more detail some of the postprandial circulating milieu, if you will.
Okay, thank you.
There's a question for Ben from Paul Morgan, University of Birmingham. So Ben, with the MYCOPROTEIN studies, what was the time period that the muscle protein synthesis was assessed post-feeding? Given the distinct difference in the relevant kinetics, would you expect to observe differences in synthesis and overall net protein anabolism if you were to manipulate the time period of which the MPS or the signaling is measured, but particularly given the high fiber content of the MYCOPROTEIN?
Yeah, I think that's alluding to a point Nick actually made. If you separate out postprandial periods, and that, when I mean separate out, I mean it's when you choose to take a muscle biopsy. So you have to be fairly selective. You can't just take muscle biopsies all the time. So you have to decide exactly what periods you're looking at. There's different ways of looking at that. You can take the kind of nutritionally relevant periods of time that Nick already discussed. So if we think we eat somewhere between every 3, 4, 5 hours, then that's quite relevant. We selected 4 hours in that experiment. If you go back to the kinetics of the two protein sources, I might have expected to see a greater muscle protein synthetic response at 2 hours with the milk protein, because you had an increased availability of amino acids earlier on. But then making that situation complex, and I guess what we've been talking about the whole of today, that's not what was seen in Nick's milk and meat experiment. So there are exceptions to that rule as well. So yes, I think that there's arguments as well about a slowly digested protein is better for longer periods of time, such as casein protein prior to sleep to provide substrate overnight. But again, a lot of those direct comparisons of those protein sources have just not been done. These experiments emerge over time, and we have various things that we don't know in terms of whether or not these data would be different if we selected different periods of time. The only thing I would add to that is I think that rather than picking at that, the field is tending to try to now translate out what it sees over an acute period to longer periods over days and weeks. And I think that's becoming quite relevant now to see whether or not these protein sources cash out when it comes to endpoints like muscle mass or protein synthesis rates over several days.
Thank you, Ben.
So Nick, we have a question here from George Pavis at the University of Exeter. He says that from the salmon ingestion study showed that despite lower plasma leucine concentration with whole salmon ingestion, FSR was as good, if not better, than your isolated nutrient condition. Do you have any thoughts as to whether whole food ingestion could cause a greater leucine uptake at the muscle level? And could this explain your observations?
No, great question. I actually just turned over because one of the individual who actually ran that study is in the room with me. I couldn't remember if we actually assessed that directly. I mean, so we're going to look at some of the co-localization work by I mean Dan Moore at the University of Toronto. So trying to see at least, can we get the mechanism there for greater, the mTOR, the whole complex up at the sarcolemma. That's, like I said, is the hub for dietary amino acids to enter. We probably won't look at, the amino acid transporters because we don't see a lot of action from in the whole muscle homogenates. They probably co-localize as well. Amino acid transporters like to put themselves around capillaries, of course. So, the short answer is I don't know if there was greater leucine uptake. I think again, with isolated protein sources, that leucine threshold hypothesis is very relevant. When we're thinking about whole foods, and I think it kind of falls apart a little bit. There's other things playing a role. What those other things are, good question. It's a great question. I have no idea. Working on it. We're working on it.
Yeah.
There's a question for both of you. So I'll ask Ben to go first, if I may, and then Nick. So ultimately, the goals of stimulating protein synthesis by adding protein post-exercise is to grow muscle mass and strength. It's pretty well established now that protein versus no protein leads to increase in mass and strength in the long term, but there are barely any direct protein comparison studies long term. Would you expect that there would be differences in the long term, Ben?
I think that there's various ways of approaching this question, and it's a sort of perpetual question for this field, really. So I think I'll approach it from this way today, which is that we do know from the longer term protein studies that the overriding determinant is that people have sufficient protein. But it's actually quite hard to pick apart relatively small effects of higher versus lower protein or protein supplemented. And the best data for this tends to be the meta-analyses of numerous studies show a small but relevant benefit of consuming greater amounts of protein. And it's essentially underpinned by the physiology that Nick described at the outset. You're looking at lesser changes when you're now talking about manipulations of very acute muscle protein synthesis rates over 2, 3, 4 hours. You're also looking at something that perhaps may not be the same on repeated meals, because that's something else that as a field, we're now translating to multiple days of nutrition. So a very unsatisfactory answer, but I would suggest that you can't always relay the initial muscle protein synthesis data to long-term changes in muscle mass, but it certainly gives you clues on where to look, because that's how we've learned about what happens long-term, by understanding the initial mechanisms of protein turnover.
Thank you, Ben. Nick, would you like to add to that, please?
No, I wouldn't. Just kidding. No, I mean, so first from an acute MPS standpoint, it's hard to chase a long-term outcome, because remodeling, or protein synthesis is a remodeling marker, and it comes in all shapes and forms, right? You can have hypertrophic remodeling, non-hypertrophic remodeling. We know generally turnover is good, of course. So generally when you're running a training study, you're going to try to view it in one particular lens. But again, somebody else could be they're remodeling, but maybe it's not in terms of just accreting a ton of myofibrillar proteins, but instead, they're just remodeling the tissue, which is great. And the second answer, I think, was what Ben alluded to. Those studies are very hard to run in terms of these. It would have to be a controlled feeding study where you're giving everybody every meal. And humans are humans; they do what they do, right? You try to control these long-term studies as best you can, of course, but they just they're still variable. So I just think the translation to me, it makes a lot of sense. When it doesn't, again, it's a remodeling marker, and again, to run that kind of study would be real challenging, in my opinion. But I agree with Ben as well. The acute MPS measurements, it's a compass. Turning on turnover is good, but like I said, everybody's different. How that turnover response might equate to an adaptation.
I mean, further to that, if I may ask both of you, what about protein breakdown? I mean, we're talking about MPS. What about, do you envisage differential effects on protein breakdown? And I mean, I guess related to that, presumably there are differences in insulin response as well to those different sources of amino acids, which may impact upon both parts of the turnover, protein turnover.
Sure, yeah, I can. I mean, breakdown. I mean, I always, protein synthesis, of course, is a little easier to measure, at least at the muscle level relative to breakdown. And I'm fairly lazy, so that's why I just focus on protein synthesis. But no, I think breakdown has a role at some level. Again, though, I think we have to look into all the data that shows us. And recently we've been looking at FBR, or direct measurement of muscle protein breakdown. which can be challenging because the modeling gets a little hard when you got these exogenous amino acids coming in. And I always say when the modeling starts to overtake the physiology, I think that's always, you know, kind of a challenge, or you question, what am I actually looking at? In short, does breakdown play a role? I think that's a good point. Yes, of course it has a role. But again, even if we allude to, I guess Stu Phillips has his recent big cell metabolism paper where he pointed to, again, you know, synthesis was a primary variable changing some of the adaptations they were seeing in those studies. I guess until we get some better methods to look at breakdown, you know, I don't have a great answer, which has been consistent with today.
Kostas, I'll just chime in on that. I mean, we see with, for instance, the mycoprotein work that we see a much bigger insulin response because we're just feeding with some carbohydrate. So I guess what we're all aware of is that there's a fairly good literature base to suggest that you do need an insulin response to protein to stimulate protein synthesis rates. Perhaps doesn't need to be very big to facilitate maximal protein synthesis rates. So usually some isolated protein will achieve that. On the breakdown side, I think there is some work to suggest that an increasing insulin will have a bit more of a dose response effect of inhibiting breakdown. But at the same time, at the whole body level, relatively small protein meals seem to have a maximal effect on suppressing protein breakdown. So I agree with Nick. I mean, I do think we need more data on it, but I think that we've got reason to believe it's less predictive in healthy individuals. The other caveat to that, though, is, of course, it talks back to the previous question about whether or not the acute protein synthetic response predicts the long-term effect. More data from Stu Phillips, which was a nice translation from the acute to the longer-term measures of protein synthesis with D2O, demonstrated that once you got past the initial damage side of unaccustomed exercise, those acute responses were really predictive. And of course, the damage side is associated with that really high rise in protein breakdown. So there's definitely really interesting work to do to get to the bottom of that, certainly.
Thank you both.
So a few more questions now. This one is from Alex Hughes from King's College London, and is for either Ben or Nick. Is there much research into the muscle protein synthetic response in males versus females when ingesting whole foods / the optimal dose of protein required to stimulate muscle protein synthesis in males versus females?
I mean, I can just speak briefly on that. I mean, in our most of our experiments, we use both males and females. Of course, we're going to be underpowered, I guess, to really chase that question directly. You know, in our experiments, we don't see any differences. Some of the larger scale work that Stu Phillips has done with Dan West as the lead, we've shown that after exercise, again, this was with whey protein, we didn't see any differences in the response, either at the short term or longer term. So, I mean, interesting point. We haven't noted any, I guess, is the short end on our end.
I would concur. I mean, I think we've got some good data on whether or not there are sex-based differences. Moving on to how those relate to the more specifics, I think we're some way off that.
There's a quick question for Ben. Could the differences in the energy content of the mycoprotein explain the greater muscle protein synthesis, Ben? Potentially,
yeah. I mean, I think there's a few studies out there that give us reason to believe that perhaps when we test individuals in this very laboratory controlled situation where they're fasted overnight and we feed them a relatively low energy meal in terms of an isolated protein, that perhaps some of that protein is just required for just general energy metabolism, and therefore providing a preferential substrate like fat or carbohydrate might allow more amino acids to be available for protein synthesis. But, you know, as Nick's pointed out before, we get a proxy of this by looking at amino acid oxidation. And then those studies where I guess the truest test of that, where you just simply provide additional carbohydrate or additional fat, don't seem to augment the anabolic response, even when you don't perhaps have quite the optimal amount of protein. So I think it's possible. I find it difficult to draw that as a direct conclusion from the literature that I have available, but I would certainly not close the door to that.
Ben, I mean, just if I may, you mentioned fat there, coingestion of fat and glucose, and I think Nick in his talked about some data from Maastricht. But what would happen? I mean, that's obviously co-ingesting fat and carbohydrate with protein. What will happen if you ingest the fat or the carbohydrate before or in the hour before, the hours before? And I'm asking that because we've published a paper recently with Luc van Loon and Francis, and we've shown that if you ingest a fat meal 4 hours before, you can actually double the muscle protein synthesis to protein meal 4 hours later. So I was wondering whether there's timing involved in terms of how fat affects protein synthesis. Could you comment on that?
Yeah, I mean, the fat it gets murky because you also then can start looking at whether or not a high-fat diet or repeated high-fat meals, so there's definitely some conflicting literature. I mean, I think I would just lean on what I've just said there. Is that I think two aspects. I think I would lean on the fact that we don't know whether or not there's a beneficial effect of providing an additional energy substrate so that we can use the protein more effectively. But we also, I mentioned it earlier on, we've not got too much information on how subsequent meals affect one another. So Nick and I did a study when we were both in Maastricht together in Luc's lab, where we tried to start picking at this, where we looked at subsequent protein feedings and to see whether or not one high protein meal affects the response to another protein feeding meal. And I think we're at the start of learning about that. Luc has nice data on habitual protein adaptation, but things like how fat interacts over several hours, I don't think we know too much about it. I think there's a lot to do there on the timing of it.
Thank you.
So we have a question here. I think this has probably already been addressed. This one's from Brian Carson, University of Limerick, for both of you. Though you observe differences over an acute 5-hour period, where a single food source or isolated protein is used, do you think this will translate to a free living situation over time in terms of muscle mass? I think we've probably addressed that.
Yeah, I think we have.
In which case, there's one here from Ollie Whittard from King's College London. Another question for both of you. Where are we in terms of a DS as an assessment of, quote, protein quality in light of the new data you are generating?
In terms of diet protein quality score, I guess that was the question. I mean, DIAS, I think, I mean, we do a lot of DIAS work here at the University of Illinois, so I'm pretty familiar with the method. I mean, DIAS, you can't do with exercise. What I struggle with the DIAS scores is that essentially they just discount the notion that exercise impacts digestibility. I mean, think about this. If we can manipulate physical activity, just about impacts every other nutrient. Why wouldn't it? Protein. So can we manipulate diets by prior exercise? I mean, they can't shove these pigs on treadmills. I mean, it's just the nature of the method. I think DIAS is good for what it does, but I think we need to couple DIAS with other measurements as well, as I've alluded to in some of our reviews that I wrote a couple of years ago. You know, just two different things. I think DIAS is good. They say they're doing what they do. It's hard to do in humans. Can't look at the exercise interaction. I think is really important. I don't think we can live in silos. You can't really have a nutrition guideline without an exercise guideline, or vice versa. There's such a synergy between the two that you have to couple them. And I think we got other methods that we can apply in humans and do them quite well and get some good answers.
I would concur with that, and just to reinforce the message that we have to look in vivo in humans, and perhaps just add that we know we can take the same protein source and see different plasma amino acid kinetics. So, for instance, another paper from Luc's lab came out a few weeks ago, just demonstrating that the food processing, for instance, affects the plasma amino acid profile, and then perhaps also the anabolic response and things like that. So I think, you know, most of these things are not going to be encompassed within a DS score or necessarily a PDCAS either. So I think we just need to reaffirm that we do need to do these experiments in vivo in humans.
Okay, so Kostas, I think that's it, isn't it, in terms of questions and discussion and time? Do you have anything else to add?
I just wanted to ask you, both speakers, a quick question about the merit of having protein from plant sources versus animal sources for kids that grow. So, for example, during puberty, there's some evidence that animal source protein might be detrimental if it's an excess in terms of fat accumulation. Can you comment on that topic, if you may?
And actually, if I could just chip in there, there is another question here that is, what type of protein diet would you recommend for protein synthesis in developing countries? So perhaps you could also address that in the same answer as well.
Yeah.
Yeah, I mean, nutrient overload, I think, is negative in any sense. I mean, so yeah, I would say that not eating a varied diet is an issue not only for kids but for adults. In developing countries, I mean, of course, that's why I'm always arguing about the protein RDA. I mean, protein RDA has been great. It's saved a lot of lives, right? In many countries, developing countries as well. In these developing countries, that's when you just have to leverage, complementary plant protein pairings. An egg, high quality protein source that can save, or really help with obtaining protein requirements. You just have to be a little smarter, like I said. If you only have access to plant-based proteins, and like I said, you're going to have to leverage the complementary protein pairings to increase the protein digestibility, as well as making sure you're getting your amino acid requirement.
I think it just underlines that we need those data on multiple sources, because protein choice for some people tends to be, what is the best protein I could possibly have at the best possible time, the best possible amount, and I will do that. With other people, it's what's available, and it's not necessarily for an optimal. It's for to be sufficient for an RDA, for example. So I think that the requirement for us to develop a broader database across protein sources, so we know what we're dealing with, and that in turn will feed into an RDA, which may or may not be different depending on the protein that people have access to. We already, as a field, talk about trying to alter RDAs depending, on exercise or age. There's no reason in the future why that might not change depending on where that protein's coming from. But I think we're a bit far away from that. And at the moment, my major concern would be if somebody's getting sufficient protein from anywhere, would perhaps be one way of answering that.
Thank you both. I think there was a final question that was actually related to that, about growth in kids. I think we probably have addressed that question. Francis, do you have any more questions, or?
I think the one thing that struck me from both talks, I think it's already been addressed, is this idea of whether plasma leucine is important or intracellular leucine or something else that is important. That's the one thing that stood out from both talks for me. And also from both talks was this idea of the role of fat. It seemed that particularly the egg study, which I also thought was awesome, Nick, fat seemed to be one of the main sort of differences there, similarly perhaps with the salmon, and then also perhaps in some of the mycoprotein work. And I'm just wondering if there's anything I'm not sure if anyone's looked at the level of metabolism within the muscle itself. Is metabolizing fat within the muscle affecting branch chain amino acid metabolism, for example? There's several pathways where they converge, perhaps at the level of, certainly within the mitochondria, at the level of the branch chain keto acid dehydrogenase complex. But that may influence how the muscle handles amino acids, and that may affect intracellular amino acid concentration or availability. And if you have any comments on that, or is that just work that still needs to be done?
My comment is, Francis, you got a lot of work to do. I think those are great concepts. We don't know enough. Like I said, I mean, we generally, for years, at least in the U.S., we were fat scared, and now that paradigm's been shifting a little bit. I think there is something with the lipid-rich food matrices. I just don't know what, but yeah, I agree that you have a lot of great points that you underlined. There might be some work out there on them, you know, at least from an isolated perspective, but certainly not from a whole food matrix. At least I'm not familiar with that work if it does exist.
Also, perhaps a kind of nexus for trying to understand the competing role of lipid, because we also know under various conditions that lipid overflow is inhibiting protein synthesis. So perhaps looking at it at the level of the muscle and potential counter-regulation of mTOR, for instance, would be the way to start picking apart that.
Right. Thank you.
Yeah, I think you have some closing remarks, Kostas, but before you do, I'll just once again thank everyone for attending today, and in particular thank our speakers for some excellent talks, and also the Physiological Society for allowing us to run this seminar. And I'll pass over to Professor Tsintzas as one of the leads for the Human and Environmental Exercise Physiology Special Interest Groups.